First of all, I'll introduce the recipient of the medal lecture. It's Dr. Michael Danielov. And he began his research in the 1970s on pathogenic mechanisms and thanatogenesis of post-aggressive reactions of living organisms. So the research revealed that the best approach to future medicine would be to start using substances that naturally exist in the body instead of creating and administering new molecules. With his team of science, he discovered methods of copying nature, actually learning how to put together multiple biological informational substances in physiological concentrations, that's nano and pico quantities, that are used by living systems to repair malfunctioning lines of information, communication, and to help preserve their biological stability. So based on his study, Dr. Danielov developed the technology of a delivery system that is the imitation of a cell membrane. And within this cell membrane, he's been able to incorporate active molecules which could be delivered to designated places, skin being one of them. With this, Dr. Danielov opened a new chapter in fundamental science, later on defined by him as life science nanotechnology. It is my pleasure to welcome Nana to present this year's SCS Medal Lecture on behalf of Dr. Danielov.
You know, there comes a point in history and time when we just take a pause, and when certain individuals who have toiled and worked very hard make a difference. Things that we take for granted, like electricity and modern medications, have all come from certain fundamental discoveries that reshaped how we then think about medicine and future technologies that we produce forward. This is one of those moments. What we're going to learn today is not another scientific fact, but it is a new direction that is going to allow everybody to re-strategize how they focus on further treatment possibilities. And let's get to know the creator, Michael Danielov. Michael Danielov is an MD and PhD in pathophysiology. Grace Abamba already gave a little history. I'll just kind of give some backgrounds. Dr. Danielov in 1978 founded the Laboratory of Radionuclide Microanalysis of Hormones and Biologically Active Substances in the Institute of Experimental Morphology. He was the chief consultant for the Academy of Science for the former USSR and led projects in the radioisotope field. He's the author of more than 40 openly published scientific articles and one book in his field. Based on the 25 years of scientific research and working in harmony with the skin and body, Bionova was founded by Dr. Danielov and his colleague Dr. Sepper, who is the head of R&D. Bionova today is a New York City-based lab where we utilize life science nanotechnology and the proprietary technologies of NuCell Direct, our delivery system, and OptiPath, which we'll get to know in a second. And based on our two proprietary technologies, we have been producing skincare, nutritional, and personal care treatments in those categories. And you have 3 stages here. First, level formation of signal. Second is the identification of the information. And the third is the transformation of a molecular signal into a biochemical reaction. Okay? So this is BIT. BIT is how our cells communicate. And that is what the scientists have discovered and learned over the course of years. And this is the process. OptiPath is a method of bionutrient composition in nano and picogram quantities. Okay. So, by imitating the biological informational transfer, BIT, we are able to support and enhance the self-healing and self-repair process when there is a dysfunction. Select appropriate how? Select appropriate bioactive substances indigenous to the human body. Combine multiple complexes from selected bioactive complexes and provisionally stabilize them, and combine the complexes into nanomodules with 3 identifiers: first-degree messenger, informational identifier, transmembrane factors, intracellular transmitters that make up the structure of BIT. And this is what brings us to NuCell Direct and why NuCell Direct was created. So NuCell is the precise imitation of the human cell membrane. That is our new delivery system. NuCell is an empty delivery system for bioactive substances and OptiPath nanomodules. NuCell Direct can be formulated for all forms of applications, topical and enteral, and it has simultaneous action to normalize dysfunctional cell metabolism, and it helps to restore bit with active action of multiple active signaling substances. This allows to reduction of side effects, the time-release targeted tissue delivery. NuCell can be formulated with controllable time-release effects and to also target specific tissues. NuCell allows to encapsulate active substances in nano and picogram quantities, which prior has not been available to us before, and ensures their delivery in that quantity dosage. And very important, shelf life. Nanocomplexes or any combination of active substances combined into NuCell can be freeze-dried, allowing for long-term storage. In comparison, liposomes are unable to go through the freeze-dry process and usually are in liquid form, limiting their shelf life. Liposome has done a lot of great things, and technology does move forward. And this is what we did, and this is Michael Danielov's discovery, NuCell Direct and OptiPath. And we're very excited to share this technology with people, and we're very excited from the results that we have been providing everyone, and we open the discussion for further applications with everyone here today. Thank you.